Hello everyone, welcome back to Studying with Zahra. This is going to be part 2 of our AQA Biology introduction to carbohydrates. Specifically, we're learning about essentially just like the basic units and everything. So, last lesson we ended off with monosaccharides, so I'm going to carry on from there. And we've done most of this stuff in GCSEs, so I'm going to go over it really briefly, but I will still go over it. Now, we're going to be doing the test for reducing sugars. So all monosaccharides and some disaccharides are reducing sugars. Reduction is essentially the process, we learned about this in chemistry, but it's a process by which a substance gains electrons or it gains hydrogen. Oxidation is the opposite, so you lose electrons or you lose hydrogen, or you could also gain oxygen. But that's not what we're going to be referring to in this specific scenario. Now, reducing sugars are substances or sugars that essentially donate electrons or donate hydrogen, allowing another substance, in this case Benedict's reagent, to be reduced. Now, Benedict's reagent is essentially copper 2 sulfate, which is blue in color, and this is reduced to form copper 1 oxide, which is red in color, upon the heating and adding of whatever reducing sugar. And so we can see here, the test is pretty simple. You just add the food sample to a test tube. If it's not liquid, then you have to grind it up. Add it with your Benedict's reagent. You can add equal quantities. Place it in a water bath for around 5 minutes. And then observe the color change. And the color change varies depending on the concentration of the sugar. So if you have a lower concentration, it's going to be like green, maybe yellow. Higher concentration goes into brown or red. Okay, perfect. Next we have carbohydrates, disaccharides, and polysaccharides. So a glucose joined to a glucose is essentially a disaccharide known as maltose. Lactose is made by a glucose and a galactose. Fructose and glucose make sucrose. And those are the 3 main disaccharides that you need to know of. Now, before we get into the nitty-gritty, something that you do need to know for sure is the fact that glucose has 2 types. You have alpha glucose and you have beta glucose. Alpha glucose differs to beta glucose in the sense that the hydrogen is on the top, and you can kind of see it here. Whereas in beta glucose, we can see that the hydrogen is at the bottom. Monosaccharides join via a process known as condensation in order to form a disaccharide, resulting in the loss of water molecule. That's pretty much just a revision from last session. Now, the water needs to be added under suitable conditions in order for the hydrolysis reaction to occur, in order to break the glycosidic bond between the 2 monosaccharides, specifically when talking about carbohydrates. So over here we have a little diagram. We have 2 actually. And the first one shows the formation of a glycosidic bond. So you can see over here we have OH from the left, hydrogen from the right, binding together, forming the water, and so the O from the 2 alpha glucose join together, forming maltose, which is going to be our disaccharide. And over here is literally the exact opposite. And you have to bear in mind that alpha glucose is used for maltose, as well as most, or just essentially just like storage molecules. For non-storage molecules such as cellulose, which is specifically in plants, you're going to have beta glucose, and we'll learn more about that in a second. Now, the test for non-reducing sugars is a little bit more complicated, but not to worry. So, for example, sucrose is a non-reducing sugar, unlike, I think it was maltose that was a reducing sugar. That was also a disaccharide. Now, they don't change color when they're added to Benedict's reagent, and the reason behind, essentially the reason, or the only way that we can, test for them is by hydrolyzing them first into the monosaccharide. So for sucrose, that would be hydrolyzing it into glucose and fructose, which are the monosaccharides. Now the process carries out as follows. So if the sample is not already in liquid form, it must be ground up. Pretty logical, I suppose. We add 2 cm cube. It really doesn't matter the quantity, honestly. Just say equal quantities of the food sample to Benedict's reagent. Test, heat it up in the water bath. If no color change is observed, then we have to do the following. So you're going to first make sure that no color change is observed, because it's such a wastage of time if it was a reducing sugar and you've gone through all this process and you could have just done the reducing sugar test, right? So if it doesn't change color, then a reducing sugar is just not present. Add 2 cm cube of the food sample, or equal amounts of the food sample, to dilute hydrochloric acid in a test tube, placing it gently in boiling water for 5 minutes. The HCl will hydrolyze any disaccharide present into its constituent monosaccharides. I've made a little diagram here. So this is our test for those specific sugars that aren't reducing. So we're going to exclude maltose or any monosaccharide from this test. And once again, the reason why we can't figure out, or we can't, do this test with the Benedict's reagent is because these sugars are just non-reducing, and so in order to kind of figure it out, you have to do this specific test. So step 1 is you grab your food sample. In this case, I have bread, which is really random, but you're not going to have bread. In case it's a solid, mash it up, put it in a little sieve, obtain 2 centimeter cubes of the sample, add it to 2 centimeter cubes of Benedict's reagent in a boiling hot water bath. No effect means it's a non-reducing sugar. By no effect, I mean no color change. So step 4 is we're going to add it with equal quantities of hydrochloric acid. Once again in a water bath, hydrochloric acid hydrolyzes our sugar, the non-reducing sugar, and it splits it up into its constituent monosaccharides. So for example, if we have sucrose, that would be glucose and fructose. And then we're going to add sodium hydrogen carbonate to our HCl sample, and you are going to test with litmus to ensure that it's alkaline, because Benedict's doesn't work in an acidic solution. And then you're going to do the Benedict's test. Now, I know this seems really overwhelming and really complicated, but really it's not that bad. I'll break it down. So Benedict's test, we already learned about that, so oops, my bad. Don't know what happened there. But essentially, perform your Benedict's test, no color change. You're going to add equal amounts of your sample to equal amounts of hydrochloric acid in a hot water bath. This hydrochloric acid should hydrolyze the food sample, breaking it down into its constituent monosaccharides. Once that's done, we're going to add sodium hydrogen carbonate, and then we're going to test to ensure that the sample is alkaline. You can test it using litmus paper. It should turn, I'm pretty sure it turns blue. I hope that's right. Correct me if I'm wrong. But then you just perform your Benedict's test once again. And yeah, that's pretty much it. So, not too complicated. Don't worry about it. You'll get the hang of it if you read it a couple of times. So yeah, add in your sodium hydrogen carbonate, neutralize the HCl. It needs to be alkaline. Retest it with the Benedict's, and it should turn orangey brown as a result of the fact that there's a non-reducing sugar. So next we have polysaccharides, and we're almost done with this little subunit. And then I might make a separate video for starch and the storage molecules, essentially. And then we're pretty much done with carbohydrates, which is really, really easy, honestly. It's 3 lessons of like 20 minutes. And I feel like the introduction is really good in the sense that it's very simple. And even if you don't find it simple, trust me, read it a couple of times. I promise you, you will get the hang of it. So in terms of polysaccharides, we know that they're polymers, and the monosaccharides are joined to form polysaccharides as a result of condensation reactions, resulting in the loss of water molecule as well as the formation of a glycosidic bond. Once again, I do want to reiterate this again and again and again because it is such a crucial and important concept that you need to grasp. Because polysaccharides are very large, they are insoluble. This feature makes them suitable for storage. And yeah, when hydrolyzed, the polysaccharides break down into disaccharides or monosaccharides. Some polysaccharides, such as cellulose, are not used for storage, but are used to support the plant cells. And I did mention before how cellulose is actually made up of beta-glucose as opposed to alpha-glucose, which is used for starch and glycogen, specifically the storage molecules. We'll learn more about that and specifically why a little bit later. But all you need to know right now is polysaccharides are very large, they're insoluble, and as a result, they don't affect the osmotic gradient. So we're going to focus on the hint right here. A few basic monomer units can be combined in a number of different ways to give a large range of biological molecules. And we also learn about this with amino acids. There's only a few, I think there's around 20 types of amino acids, or around 20. However, there's so many types of polypeptides, and the reason behind that, or so many types of proteins, is because there's different combinations, different ways of foldings, and just different proteins as a result, I suppose. So yeah, over here it just kind of shows the breaking down, polysaccharide into your disaccharide into the monosaccharide, and so in each one of these, so if we focus over here, I have my two polysaccharides, and they are broken down via the addition of a water molecule into a disaccharide, and then another water molecule turns it into a monosaccharide. So 3 hydrolysis reactions have actually taken place, or sorry, 2. To go from the polysaccharide to disaccharide and the disaccharide to monosaccharide. And so 2 glycosidic bonds are broken down. Starch is a polysaccharide that is found in many plants in the form of small granules or grains. So for example, in chloroplasts, you have small starch grains. It is formed by the joining of between 2 and 100,000 alpha-glucose, molecules joined by glycosidic bonds in a series of condensation reactions. So once again, starch is a polysaccharide. It's a storage molecule. It's found in little granules and grains, specifically in like the chloroplast. It is made up of many alpha-glucose molecules binded together via glycosidic bonds in processes known as condensation reactions, resulting in the loss of a water molecule. And yeah, that's pretty much it. Next, we have the test for starch. So starch is easily detected by its ability to change color in iodine. This is a really simple test. So essentially, you have iodine, which is normally orange-brown. You add a little bit of your starch sample, suppose equal quantities, or actually you don't need to do that, honestly. Just add 2 drops of iodine to your little sample. It should turn blue-black. And you have to shake it or stir it a little bit. So for Benedict's, heat it. For iodine, shake it up. Okay, we're going to be doing these summary questions, and then we're done with this session. Identify which one or more monomer units make up each of the following carbohydrates. So for lactose, we have glucose and galactose. Sucrose is glucose and fructose. Starch is made up of multiple alpha-glucose monosaccharides that are joined together by glycosidic bonds via a result of many condensation reactions. Glucose combines with fructose to form the disaccharide sucrose. From your knowledge of how disaccharides are formed, deduce the formula of sucrose. Now, at first thought, it might be easy to think... Oh, by the way, glucose and fructose do have the same chemical formula because they are isotopes of each other. So essentially what that means... Sorry, they're not isotopes, they're isomers of each other. Essentially what that means is they have the same chemical formula but different structural or displayed formulas. And if you don't particularly get that, maybe watch a couple of videos on chemistry GCSE, specifically isomers. We did briefly go over this in organic chemistry, so let me know if you guys want a separate video on that. But essentially, your first thought might be, let me just add them together, right? So C12, H24, O12. That's probably what you think at first, but that isn't exactly the case. Now, we know that in order for the glucose and the fructose to bind, because they're both monosaccharides, a glycosidic bond must be formed and a condensation reaction must take place. So a water molecule is actually lost. So instead of doing C12H24O6, you're actually going to subtract, we know a water molecule's formula is H2O, so you're going to subtract 2 hydrogens and 1 oxygen. So you're going to get C, instead of 24, you're going to get 22. And O, instead of 12, you're going to get 11. And that is a little bit tricky, but just remember the fact that a water molecule is lost. And that water molecule is lost from the monosaccharides themselves. It's not just appearing out of thin air. To hydrolyze a disaccharide, it can be boiled with hydrochloric acid, but if hydrolysis is carried out by an enzyme, a much lower temperature, 40 degrees Celsius, is required. Explain why. First of all, I just want to mention, you might be wondering, how is hydrolysis carried out by an enzyme? Well, you know how we learn about our digestive system and everything, and how, for example, maltase is used to break down maltose into the constituent glucose monosaccharides. That is also a hydrolysis reaction, and you might not have thought of it at first, but it is pretty much the breaking down or the hydrolyzing of the disaccharide into a monosaccharide, which is pretty much what a condensation reaction is. A hydrolysis reaction is. And so we learn about how enzymes tend to denature at a high temperature because the bonds are broken and the shape is just meddled with. The active site is no longer complementary to the substrate molecule, and the enzyme just denatures. So that is pretty much the explanation. Once again, over here, you add your iodine and it turns a blue-black. That's actually a lighter shade. It generally is a little bit darker. But that's pretty much it. We're going to be doing 1.4 in the next lesson. Once again, let me know if you guys want to see anything else for any subjects: physics, maths, chem, bio, sociology, GCSEs, in general, studying tips. Just let me know, and I will gladly post. Thank you so much for watching, and I will see you guys next time, hopefully.